We're not even interested in cells. I mean, we're interested in DNA. DNA is not living, right? So we've passed from the notion of, I mean, in Foucaultian sense, he thinks about, you know, when we move from natural history to biology. It's the beginning of, biology is about the study of life itself, right? We're beyond the study of life itself. If anything, we've grown kind of unconcerned with that, and what we're really fascinated with are these these micromolecules inside of living things. So we've gone back to a kind of, I think we've left biology at this point.
Genuinely love it.
So I can hold it down.
It's good to be here.
Oh, here comes
when we shut this down for the night, when we're leaving this up, we need to remember to charge the drill battery.
Let's spark this place up.
All right,
Let's see what it's got. See what this lab's made of. So this is it should be what's called LB, Luria broth. Nice stuff for E. coli to eat. And it also turns out that the TAC bacteria from my studies have seemed to thrive on this as well. This is a vitamin stock specifically for Thermus aquaticus, which is the bacteria that we're hoping to isolate here. So we want these things not only to be able to eat, but to be able to breed. And so to do so, we need the right amount of foodstuffs for it to eat, and the right amount of vitamins and minerals for it to, that also needs to survive. TAC is a pretty interesting bacteria. It's not just your standard deal. It's a long, filamentous thing. It looks like a giant snake of the, or python of the bacteria world. It's got a flagella at the end so it can even move around and make it smell a little bit like the center of the earth. I'm afraid my bacteria is pretty unisex, as far as I know. There's some have written about the fact that it can also be a spore formulating organism, so it can have a kind of 2-cycle lifespan, but I don't understand that part of it. All I know is what it eats and where it likes to breed in. And I want to get the real classic looking stuff. I don't want the spores. I want the real filamentous buggers. I always find that the most important thing with all this biology stuff is just to be pretty organized about what things are which. You want to assist me?
Yeah.
Basically, I'm just trying to keep this stuff upright while I'm pouring. So maybe, do you mind just holding, or after can I just pass you a tube when I'm done with it?
Yep.
I'm quickly preparing, adding samples and preparing its nutrient base. I've just added 5 microliters of sample, and now I'm adding the remainder, another 5 microliters of the vitamin base that we were talking about. And we're going to add another 20 of this stuff, which is basically the food base. This one particularly, what you want to do is you want to only have it open for a split second, because 1 or 2 spores of fungus get in there and it's all over for this whole mixture. Could you please pass me number 4, please?
Number 4?
And if you wouldn't mind giving it a little shake before you unscrew it. Why, thank you.
You're very welcome.
Lovely. I must say I could not have shaken that one better myself.
What's going on here?
She's shaking and unscrewing my bacterias. Sometimes
straight from the sack.
But mostly she unscrews them.
I'm not sure what's going on at all, really.
What this would be is sort of some water samples.
Okay.
Which might have a little squirmy little fellow in it called Thermus aquaticus. And we're looking, this is a search for Thermus aquaticus. Okay. We might just be able to get a little thermos to start growing up in our tubes. Number 6, please.
Number 6.
Yes.
Gently shaking.
The process of DNA amplification via the polymerase chain reaction process involves needing to heat samples up to 95 degrees Celsius to force the DNA strands to denature. So the Thermus aquaticus bacteria, particularly a polymerase that it creates for copying its own DNA, is thermally stable at these very high temperatures. And so it's that quality that allows us to produce, when we amplify DNA, literally billions of copies. The reason this is relevant to the area is that the hot springs in this area are where this kind of bacteria is native. Originally, they were isolated in Yellowstone National Park, but they've since been found in many of the hot springs around. We're going to see if this one works as well as the kind of classic Yellowstone variety. Yeah. But we're in this kind of setting. We're going to try to, go back in time, stir them up, mix these little buggers up, and see if we can't get them to assist us in amplifying some human DNA. At some point, they get saying, well, how exactly do we go about making art with this stuff? So yes, if you would give number 1 a nice little shaking. Excellent.
So the shaking's just in case it sediments to the bottom and mixes it up?
I just like to see her shake.
Straight from the sack.
Straight from the sack.
Do you have a proper garbage yet?
Not yet.
No, okay, I didn't think so.
You got your cowboy scarf?
This is an incubator.
Come here.
And we should be able to set it for whatever temperature we need. We'll probably have to monitor it a little bit, but we want to take this all the way up to 70 degrees Celsius.
For how long?
For a couple days. They like to be kind of shaking around a little bit because
in the womb or in nature.
Kind of, yeah. I mean, basically everything with these things, they need to be able to get air, and so they need to be able to kind of get sometimes recycled to the surface. We'll see. We'll wait to see about the film.
Just wait out and the moose kind of sit there.
Just a matter of waiting. It's still quite early. It hasn't even been.
And the guys go like this.
Only been about 12 hours of actual heating. So we'll see. We'll see what happens in there. I got a better idea. I could use is something to spit this into. I'll be right back. I'm just going to get it. You don't want such a thin opening, and I'm going to pipe it into each one of these vessels. One of these. 2.35 grams. Should be exactly what I need. So I can use standard table salt. Here we go. 2.3 should be fine. This kind of disrupts the loosest dead cells in your mouth. Let's just shake this up until nothing's on the bottom. Something just disgusting about holding salt water in my mouth. Damn. Ugh.
They're getting bigger in the lab.
Yeah, Jesus Christ. As long as, I mean, this is a little bit disgusting, but I mean, it's only my DNA. It's only my spit after all. You don't want to have one tube much less full than another. I think that's gotta be fine. To those and these two. Should be pretty similar heights too. So then these are going to go into the. These are going to go in the centrifuge. I'm assuming full speed is 1400 RPMs. The centrifuge on there's a little bit more fingers in the spit than normal, but it's,
what are you gonna do?
You know what you gotta do.
We're in the field.
We're in the field, man. You know. Now I think from this, after this point, is basically when we're going to want to get that fire going again. I actually got a little further than I was remembering I wanted to go to.
Okay.
That's spit stuff, so nobody should drink that.
Okay. When you're done with it, you can put it in the BioArtCamp container.
Actually, it'll save it for now because we can. This little pellet there, you probably can't see it, but that little pellet at the bottom are the squished up cells. What you kind of do is you flick the little stuff around there, or resuspend it into the fluid so that it's not so absolutely compressed. Yeah, good. And usually I actually hit this. Sometimes the really good centrifuge can be a problem because it actually mixes them too well. Okay, that's done. I'm squirting them. I'm trying to dissolve them as best as I can in that leftover water that's in the bottom of the tube, because we don't really want them to be too clumpy. They're a type of little molecular beads that I'm trying to remember to explain how they work now. Well, they're kind of miraculous, because basically what they do is they allow me to, in one step, to get the DNA out from it allows me to extract the DNA basically in one step from my mouth, which just involves various heatings on the in my waters. When the cells burst, it will stick to all the cell membranes, but it won't stick to the DNA. And so that when I finally pull this stuff out of the hot water baths, I should be able to like spin it down in the centrifuge again. All the cell membranes and crap will go to the bottom, and then the DNA will be left in the fluid, and that fluid then will be what we're looking for.
Because you have a sort of MacGyver-like approach to your equipment.
Yes, well,
Grab what you can.
I realize that we don't have forever here to make something happen tonight. We gotta. There'll be an angry mob out there if we don't make something happen. This is just. I'm just using this kit for extraction. These are my sort of more inexact thermometers. I mean, technically, I guess we're always in nature, but our special little lab area there is, I guess some would say it's in between, some would say it's continuous with nature, some would say all are continuous with nature, but for my purposes, this could be outdoors. It's only the fact of having a level table surface we need. You want to see blue? But glow in the dark fluorescent green raccoon. Is there something you can tell me?
Mama said I could eat a marshmallow.
I'm lucky. I was very lucky to have the fluorescent raccoon here, because the fluorescent raccoon here told me what I needed to know, that while all the others that were doing similar kind of things with DNA and trying to find things on the genome, they were trying to amplify the signal. They kept trying to figure out how to make DNA glow more brightly and like making better radioactive probes and all these things. But what Mullis realized was that via the fluorescent raccoon was that what you really needed to do was to make more sample. Not more signal, but more sample. So then, rather than try to amplify that signal, he amplified the sample by billions of times. It took longer inside than we thought. I mean, this stuff, if you put it in a bottle, would probably be the priciest of all, you know, your natural spring waters. And this one will be the one that we use as boiling, and then this one will be the one that just gets used as you know, whatever. Just don't want you to catch a bad spark. So this one we want to get to 100 degrees, and this one we want to get to 56 degrees. And if this one gets too hot, we'll slow it down with some of that water. Keep stuff rolling.
Oh yeah.
We got some today.
Make a little mistake there, team. This is in Fahrenheit. I was off by just 20 degrees. So we'll keep those heating up for a bit.
I am also
It's simplifying everything. It used to be a very long process in the laboratory. It involved using a chemical called phenylchloroform. And phenylchloroform basically, like, it instantly vaporizes into the air, and so like instantly, like, you have this like incredibly kind of harmful kind of I don't even know what, you have this incredibly sort of harmful vaporous stuff that like is, even if you have ventilation in the room, you can't do anything else in there all day really. So this is much better. Nothing is ever perfect. When I put the cells in, they're a little clumpier than I wanted them to be, and they sat a little longer before I flicked them around than I wanted, and yeah, it's very hard to find the perfect conditions. I've yet to find those.
Now we're making laboratories.
No, but I really encouraged him to
Well,
try
I'm
his
also
best
making it with fire.
Spain can help him. Help him.
And this 56 degrees centigrade water bath. I'm going to put them into the 100, what we hope is about 100 centigrade water bath. There we go, we're at 130.
It's perfect.
So 132 degrees Fahrenheit is where we want our...
That's Ian's as well.
What I guess I like to do is to flick until they really, until everything turns kind of cloudy. It's tricky to see. I suppose it takes the most exacting lighting conditions and focus, but
No, it's all good. It's in the fire now. See there, it's exploding.
so I flick that around. When I work in my own lab, I keep incredibly close notes, and so some of these things I feel like I've improved upon, and sort of like trying to work cheap, trying to work quick, and trying to work with low-end materials. Some of them I think I've really improved. I've made some improvements upon some of the parts, particularly working with restriction enzymes.
Yeah, art for art's sake, because there's no function.
Between the salt and between the chemicals and the heat, everything should be lysed open, broken open, and the beads should be, I think holding all the cellular components with the exception of DNA. It should be binding to them. And then when we take it inside, we'll centrifuge it down, and we'll centrifuge it at a speed that's fast enough that all the cellular crud and the beads get stuck to the bottom, but not so fast that the DNA does. And so the DNA will stay in solution. So we'll suck the DNA out of the top of the tubes, throw out the bottom, throw out the stuff at the bottom, and then we'll have 4 tubes of about 100 milliliters of, or 100 microliters of decent usable DNA. Hundreds of mosquitoes sacrificed for this experiment. That one was spared. Just amplify the signal, or the sample, not the signal. That was the basis for everything in that experiment working right. And basically, it's got to take about the top, probably about the top 100 microliters of each one of those tubes, which should contain the DNA.
Here you tend to add one. Now I'm going to go for a quick hand wash.
It's been changed enough that the older machinery has all the wrong kind of information on it. Even like this fact that usually the new ones all have this conversion between the speed, the spinning speed, and the actual amount of g-forces it generates. But this one doesn't. Sometimes when you work with a lot of older equipment or second-hand surplus equipment that hasn't exactly been calibrated perfectly and whatever, you do wind up kind of doing, you kind of learn how to use those machines, and you realize that there's a big difference, or there's a difference between this idea of like what is exact and what is consistent. But sometimes just being consistent is good enough. For instance, particularly with measurements, I mean, it's provided that you're using something that's off by the same percentages each time, and you're measuring every single ingredient with it, then you know you might be scaling up or scaling down, but you're actually getting the same ratios. So what I want to do is I don't want to scoop that. All that stuff is a leftover cell mass and a bunch of other crap, which we don't want. So, but that's one tube of DNA for tomorrow. Back on. Ooh. These now should just go. We need to find the spot in the freezer for these. All right. I'm gonna try to prop them a little bit upright so they at least freeze into a.
Sorry, I'm still going to the bathroom. I don't get that.
But it's just
a home freezer.
Yeah. Sometimes I use my, if you have a tiny enough lens, on your camera, you can also do simply one of these. Looking through these puppies, you can simply. These are some of the decent images. These are some of the guys we're looking for, the tack. There's lots of little, I can maybe blow it up a little bit. You can see all those little wormy things. That's our quest. Yes. Yeah. We're looking at stuff that's like needs about at least 100 times magnification to be seen. Wow, this is... Oh yeah, this is it. This has got to be it.
So this is the bacteria?
Yeah. Can you see them, the long, skinny strings?
Yeah, but they're not moving.
No, I mean these things have been, they got cooked for probably too long, and then they sat, and then they...
So they're just sleeping, or?
No, they're dead at this point.
So take the leftovers.
Take the best ones,
yeah,
and then to like give them a bunch more food and fuel to
So some of them are actually sleeping.
Some of them could be. Where I found them, there's no possibility of sustaining this kind of life. They had come from a real sulfur spring, and now they're about to flow into a hotel and be hit with chlorine. Well, I mean, it's just like any time you do this, there's always things that are imperfect, you know, and this is no different. So I think in this time it seemed imperfect that the sample sat for more than 24 hours after harvesting, and that's not great. And we don't know exactly how far bacteria would have traveled down that spring from where they're actually really thriving. So it could be that they've been traveling for days without proper nutrients anyway. So we don't really know. So the idea here is just to try to get some quick results about if one of these is viable. Remember to swish them around a bit first. If one of these is actually viable, we're gonna say great. We're gonna harvest up the stuff from it, and we're done, right? I mean, mission accomplished. Number 7 was the one that used the smallest quantities of sulfuric salts, and the other ones all use a standard amount. So we'll see. The thing is, like, you're always operating in so many unknowns, right? Yeah, it's unknown if there's anything in there. You can't, and then you got this big unknown that nothing is visible to the naked eye, and that's what I guess I find kind of fascinating as well. Also, you can sometimes have an experiment that takes you a week to do, and you don't know what's in the end, and you're not always going to be sure even at what point the experiment failed. Methylene blue is kind of a... Yeah, methylene blue is kind of a classic. So I think we're going to try... Usually 1 drop should be perfect, and hopefully this comes out in a nice dropped form. 1 drop should be about perfect if this is a similar concentration to food coloring, I'm hoping. particularly with, ooh, that's a dead mosquito, it's not. Then you want to flick it around a lot because if Thermus aquaticus is in there, it tends to form into big matted kind of sheets, and so you want to knock it up enough that yeah, so we can see the individual ones. Here's what we're looking for.
Next one, it will be mating.
So you can see they're very definitive looking organisms.
Okay.
Right? We won't be confused by any other kind of flagellated worm-looking bacteria.
And where's this photo from?
So this was stuff I took just from a. I incubated exactly like this from a coal plant outside of Buffalo, and it was just where the coal plant put hot water back into the lake. Drop about like that. And then a piece of cover slide.
So this is where you can put the pre-made
Magnification. Well, great, now I'm...
mycelium roots.
What we're talking about, Thermus aquaticus, it could be there, and it might have just grown very strangely because of something about the circumstances. We have to make sure we have the right stuff to clean it up with. And let's try one more time. It's not, you know, nothing's gonna die. Alright. Everyone's already dead. Yeah, perfect. Okay, we'll try this next. I thought I'll be happy just to see this here and to confirm that it's here, and I can always do the other stuff in my own lab at some point. I was mostly interested in seeing if it existed. I'm just checking to see if anything grew in that solution we made on day one of the camp here. I don't think much worked. Not sure why, but I'm going to double check before I say it didn't work. Got it. Just turned out to be the lens needed a good cleaning. We can test for multiple things in this DNA, which half of it's mine, and I'll also use Adam's half. So half of it will be the DNA I extracted the other day, and half will be the DNA that...
So this is a stash?
Yeah. I mean, in this case, I'm just doing what the process is, but it's become something more... The process has become a lot more automated. So I'm just kind of doing a very non-automated version of it.
So what does that mean?
I might have to... That might be tricky to explain as I'm doing this. Oh, that's still frozen up a bit. Alright, Jamie?
Yeah.
Wiggle that between your fingers for a little bit. Two different sets of PCR reactions we'll do today.
So this is what you made this afternoon? Or?
Yeah.
Okay. So...
This, I'm not thinking the possibilities of this working are great because, for one, this is only... These primers should only amplify human DNA. So that's one reason, and the other reason is that I used a little bit more scientific types of extraction protocols, which remove all the kind of alcohols and salts from the DNA that's extracted, whereas it's hard to do that with household materials. What I found with PCRing is these. Right now, I'm doing a pretty small region of DNA. I'm doing a kind of standard size region. I'm trying to amplify regions of DNA that are about 500 to 600 base pairs long. That's actually the range is usually pretty easy. The last piece I did, I was trying to copy a famous DNA fingerprint, the DNA evidence used in the O.J. Simpson trial. And I was trying to make an exact copy of it using my own DNA. And this entailed like some really, really like heavy-duty, really exacting stuff. It took me months to get all the DNA of the right sizes because I was trying to amplify stuff that was 15,000 base pairs long. I mean, that piece is, you know, about, you know, kind of looking at the history of eugenic technologies and things, and noting kind of their overly simplistic nature, like the idea that, right, oh yeah, of course, if one's DNA moves faster, it must be genetically superior, or like yeah. It's like kind of taking very literally the idea of genetic fitness. My premise was that the molecular biology really left the door wide open for these kind of ideas to continue. You know, like for whenever you get into human genetics, you get eerily close to it, and the more I looked at it, the more I found that I was kind of hauntingly correct, in that even the original name for the Human Genome Project was originally neo-eugenics. That was part of the very name of it. All researchers involved in it realized that, like, yeah, I mean, the closest corollary we have to this is eugenics, right? It wasn't hard to believe. There's the only two phases of human genetics were eugenics and this new thing called genomics. So it was kind of like sort of all there was. You know, Shinsheimer was the one who proposed the project originally. He proposed it as new eugenics, and it was James Watson, the co-discoverer of DNA, who suggested, you know, people might get the wrong idea about us if we call it neo-eugenics. Let's call it the Human Genome Project, you know, or something less loaded. And so you see the specter completely remains. I mean, and it remains for a good reason, because it's the only, you know, it's all there ever was, right? These, let's see. I think most biologists see it as an evil specter, but they're often not as, they often don't allow themselves to realize just how close the endeavors really are, how deeply related, that there's two phases of human genetics. The first phase is eugenics, and the second phase is human genomics, and there's absolutely nothing in between, because in between there was this stuff was basically shut down by the UNESCO after the Nazis made eugenics so unpalatable.
So do you think somehow BioArt can comment on this?
Yeah, totally. I think this is, I think this is a great forum for these kind of discussions. I think in a way, like, you know, the way that interdisciplinarity is supposed to work is that, you know, people from outside related fields who get deeply passionate about the area can add their insights, and I feel like that's what I do. That's what my purpose is, and it's not that I'm the sole person adding insights, but I feel like I contribute to an interesting dialogue. I swear, like a month before I showed the piece, James Watson came out, you know, the guy who decided that eugenics was too loaded a term, and gave a talk at UC Berkeley where he said, oh, in fact, a friend of mine did an experiment in Southern America, and he injected a bunch of white college students with melanin, this stuff that darkens black people's skin, with the intention of making them more resistant to skin cancer. And lo and behold, all these male college students developed sustained and unprovoked erections. So he said, in fact, all these stereotypes of the black male and the Latin male, they're all totally true. They're literally in their DNA, specifically in the DNA that would be producing melanin, because melanin is in fact like Viagra. So in a way, you see that like these, even the kind of in the superstars of genomics, you see all these like totally latent racisms. I'm gonna put some of this stuff back in the fridge, and then I'm gonna take this stuff out to the fire.
Sure. Okay. I'm gonna change my tape.
Okay.
So it's a song about a guy who travels around the countryside breeding genetically modified pets. And his woman left him, and he's got some other problems too.
We're sorry, Mom.
And I've never played it like this, so we'll see how it goes.
I realize I'm kind of a stereotype.
Not at all.
Like a Canadian witch.
Yeah, exactly. La 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 la. Okay. We're dogging on the muggy oak trail. We're dogging on the muggy oak trail. Feel free to like howl like coyotes, like do whatever you want. You never really loved me, darling. You never did, and that's a fact. We're dogging on the muggy oak trail. I got a half jug of agar and an old PCR machine. I got a half a jug of agar in an old PCR machine. That big old jar grows the best damn critters that I ever seen. I got a half a jug of agar in an old PCR machine. I'm gonna let the acid bubble. I gotta keep that bottle clean. I'm gonna let the acid bubble. Gotta keep that bottle clean. And I never seen so many colors, not even in a dream. I gotta let the acid bubble, gonna keep that bottle clean. Oh, we're dogging on the muggy oak tree. Oh. Inspired by Paul Vanouse's artist and Wikipedia.
I was heading for Las Vegas, but I got stuck here in Reno. But I really like the music.
Oh, awesome.
What am I saying there, I guess? I think this thing's about done. I forgave for its last cycle.
PCR.
I'm still trying to do PCR in the background here. So Casey found a nice little spot down here for electrophoresis. Good a spot as any. This little jar can go here. That's going to be our what we call the tip jar. Yeah, this is the gel we made this afternoon. This is just a very standard agarose gel. And then I need the liquid here, thanks. A buffer solution. This buffer is so that I'm going to basically make an electrical field here. And the buffer is so that the field is consistent. Yeah, but see, the water looks lower there, looks higher there, right? Than here?
Yeah, it's higher this side.
Right?
Yeah.
Yeah, that looks perfect. Okay. I'll get these all set up and ready to roll. This part?
Yeah.
No, this part's not hard at all now.
No?
No, this part's pretty much just for fun. Because,
For me, it's the hardest.
but this stuff, if it's pretty standard, it's not such a big deal. Yeah, so these are the things that we, various which do it. Take some samples at least.
You're good, Paul.
Thanks.
One thing is...
Yeah.
Scientists to do art.
Couple of them though, then it was brilliant.
The sun is coming.
Maybe one more of this. I'm gonna put one more of these in there. Song is gorgeous.
Yeah.
I'm running a electrophoresis gel, which is the technical term for what is usually called, DNA fingerprinting or genetic imaging or whatever. It's just a way of testing the size of the DNA that you have. And so I'm just testing, by testing the size, how many base pairs are in my DNA samples, I'll determine a little bit about my own identity, and hopefully we'll even be able to determine something about my identity, because we did this PCR over the flames last night of the fire, and if it worked, we will then be able to tell certain things about me, whether I'm heterogeneous or homogeneous for certain kind of traits. Because one of the things I was looking at was the presence or absence of a gene called the ALU gene, and that's like a kind of a standard marker in populations, because about 1/3 of the people are homogeneous for one trait, another third are homogeneous for the other, and the third are heterogeneous, meaning that they have both. And we'll see which one I have. I don't know. And then,
can you speculate?
Yeah.
Sort
What
of,
would you like?
I mean,
What would you like?
I always think of myself as being a rather heterogeneous fellow, sort of a mixture, no purity in myself. So I would expect that my DNA is likewise rather impure. I'm a post-race kind of thinker, I think race is a colonial construct, and thus I'm against the whole notion of racialization, so in a way a lot of this work is
And how is that
positioned
working against the notion of race?
Well, in this case, this is a little more playful, because I'm doing this kind of more, I guess, 19th century kind of romantic notion of the individual. I don't know where it fits in there. There is something essentialist about that 19th century vision, but I'm not. So in a way, I guess I'm complicating it with this kind of work in this specific concept.
You're raising the problem rather than bringing the solution.
Yes, precisely. Yes, precisely. And I think of it as a different way. I really think of this as post-biology.
In what sense? Because what I was saying is was coming from Henri Atlan, who says that biology passed from being a science of observation to a biotechnology because now it creates artifacts.
I fully agree that it passed from being a science of observation. I think it passed from the science of observation to the science of rationalization. And that's why I consider it post-biology, because I think it's more informatic and statistical than it is necessarily biology.
So
just in the same.
Is like removing biology from its own.
Yeah, sure. We're not even interested in cells. I mean, we're interested in DNA. DNA is not living, right? So we've passed from the notion of, I mean, in Foucaultian sense, he thinks about, you know, when we move from natural history to biology. It's the beginning of biology is about the study of life itself, right? We're beyond the study of life itself. If anything, we've grown kind of unconcerned with that, and what we're really fascinated with are these these micromolecules inside of living things. So we've gone back to a kind of, I think we've left biology at this point. In biology, the cell was the basic unit of life, and so it's really the sort of central object of study. Now, in a way, it's kind of a byproduct of informatics, right? I mean, informatics are the basic, are what life is, right? We can strip these. We can strip a cell down to nothingness and then kind of rebuild DNA molecularly, you know, synthetically, and insert it, and thus DNA becomes now the sort of central unit of investigation. My critique is, I mean, I'm critical of this kind of essentialization. You know, I'm critical of the kind of this sort of genetic determinism that comes with this kind of strongly kind of DNA-centric science, but I'm also, I think, just pointing out here that we may have left, you know, what we formerly would describe as biology, right? We may be in a different, not that biology doesn't still exist, but the current era of big science is not biology. I think it's post-biology.
So post-BioArtCamp is more appropriate.
Yes.
So it's happening?
Yeah, it's happening. It's running a little slowly, but I think it's working. Yeah, you can see how, particularly with the samples on the left and the right, how the purple band is moving forward very quickly. You know, it's moving past the insertion point. What we're going to do is we're going to run this until the purple band is about halfway up the gel. But anyways, it's that extraction protocol is great for getting DNA out cheaply, but I need to purify it one more step for it to work in the gel.
So could we speculate, for example, if that DNA would be like from the DNA extraction it does with the food, mushrooms and everything?
Yes,
that in fact its protocol is not really extracting DNA.
No, but I fully agree that it is. It is extracting DNA, but in order for DNA to work in like electrophoresis processes or to be amplified, it needs to be free of a lot of the things that he uses to cleanse and get the DNA out. So it would need to have another step. It would need to have a phenylchloroform step, where basically you kind of dry out one layer and take the other layer. Okay. It worked flawlessly.
It worked?
Every single lane.
Wow.
I mean,
even the Adams too?
Adams didn't work, nor did the ones that came with the kit, but the ones I made myself.
Can I peek?
All, yep.
You have anything in there?
I'm gonna
Can I have a peek?
Yeah, we just need to be in the perfect darkness.
Oh, okay.
Yep. Okay. You can come and look.
Can I have a peek? You go. Just need to have a hand.
So those glowing bands are the different gene segments that were amplified, and I used very subtle variations in my mixtures, so that's why I'm getting more than one. They're very subtle variations. I was thinking maybe one would work.
And the single one is?
So this basically is just, this was a region of a skin color gene, the SRC gene.
Okay.
Hey guys, I'm in here.
Oh, I'm really sorry.
No. God, everything's happening up
Sorry, we were just looking for a dark place.
No, he's fine.
Okay.
Yeah. Oh,
and then this is a
Hey,
different,
hi, how you doing?
This is a different part of
Yes, I am. I'm way up at Castle Mountain in
this is a
hospital
different
doing
gene.
I noticed it's separated.
Yes, exactly.
From
and
where's the others are closer?
Yeah.
That one is separated a bit from it, right?
Oh, well,
no,
they move this way.
Scientists and
the gel.
Okay.
So these are all basically the same size pieces, but they're from different spots in my DNA.
And this one's a different size, the last one, no?
Petri dishes and all kinds of stuff.
These things on the side here. These are things called labs.
And
these are just commercial products that you know.
Okay. So it's just the first one there and this one.
Yeah.
I'm having lots of
1, 2, 3, 4, 5, 6 actually.
Right. 4 and then these two here.
Yep.
Oh.
If we're getting these kind of really high amounts of DNA
a few days after that, then
from over the campfire, yeah, I mean it does kind of call into question the current paradigm.
Well, I would like to say something. Sure. Seeing as I am not a scientist, I feel very educated about this whole subject matter, and I'm honored that Paul would take the time to explain this to me, because as you know, in the general public, you never get someone to explain anything like this to you, which is quite true. So thank you very much for explaining this, because you know it took me quite a while. I was listening at first, and it didn't make too much sense, and I kept listening and I kept listening. And last night I was very sorry I missed it because the foot was so stupidly bad. But so I'm happy you brought this in now. I can have a look, you know.
Because you know there's a lot of people who say that DNA is actually the essence of life, right?
Right. Yeah.
So how do you perceive self-identity in that context?
That's the kind of trope I'm playing off of, right? That we are our DNA. We don't know who we are until we send our DNA into some lab to make our DNA genetic portrait. I'm playing off this sort of contemporary trope of self. Yeah. But
so do you believe in that trope?
No. As a matter of fact, I'm probably an extreme culturist in the sense that I tend to see DNA as not being the end all of who we are. I tend to be what would be considered a nurturist, I guess, in the '60s, rather than nature is driving all our. In this case, I'm using this kind of simple, I think a idea that's very simple to deconstruct, right? We are our DNA, but I'm also kind of playing it off here in the wilderness against this kind of very romantic conception of finding the self, right? Like I mentioned before, like in Melville, like in the quest for the white whale. It's not really about the whale. It's about some other sense of who we are, right? It's about a kind of journey of discovery, but it's a journey of discovery where there's also transformation involved, right? So I'm kind of playing off this kind of romantic and this very kind of post-biological sense of self. Yeah. Right? I think the idea is just here. I mean, I even consider this now sort of towards backwoods PCR, towards a way of doing these kind of things in the deep woods. We'll see which parts work well and which parts don't, and we'll hopefully, I mean, really the idea of this project here is to have enough sort of discursive threads to pull at. So when someone asks a question about, well, why was it that it took Taq polymerase to really turn PCR from a really elegant concept into a really potent machine, right? Then we can talk about that. We can say, yes, it's because PCR could survive cycles in the way that other enzymes from standard E. coli could not. I like what I don't know, I guess Barth brings out in books, in texts like SZ or something like that, the notion that you can have an event, you can have a thing, you can have something that seemed to be really grounding of itself, but what it really is something that sits at the nexus of multiple kind of intertextual threads. And so I do tend to see things this way as well, as being sort of certain things being kind of a nexus for a lot of different different disciplinary threads, different kind of discursive threads, different cultural meanings it's coming out of. And I think this particular process has a lot of those. And I think performing this process in this space, in the deep woods, by the hot springs, has another set. Another big part of the legal issues around PCR were that the original bacteria were extracted from Yellowstone Park, the geysers in Yellowstone Park, which is a national park in the Rocky Mountains just south of here. They were never, they were taken from the national park, but the national park was never given any of the proceeds, and the proceeds were billions of dollars around these patents. So it's important to try that also in a different kind of space, and kind of to relook at that and to wonder, why is it that state parks now are very touchy about their kind of their regulations about what comes out and under what contexts and what you can do in them. And part of it is just because, I mean, we've seen all these kind of colonial practices, and in the case of Yellowstone Park, having endo-colonial practice of harvesting materials and being kind of I guess taking complete ownership of those materials, regardless of who or what they came from. So I think it's interesting being here and also going through the same set of issues, going through these sets of issues here at Banff. Where can we collect from? We have to be pretty far downstream to actually get it here because of, in this case, not because of patent worries or intellectual property worries, but because of environmental ones. So this day and age, the reason we can't collect too near the spring itself is because of an endangered slug, or endangered snail, which is also thermophilic. And it's interesting to see, again, how these things kind of play out. Maybe the experiment will work or not, because of the presence or absence, because of the presence of the snail. I tend to be interested in this notion of how we do identify something, and I think even my processes are more focused on the laboratory than necessarily on the organism. Like, I'm really fascinated with the way that these laboratory processes deconstruct identities and things like that. I think the space of sort of all the kind of contradictions of what a lab is, right? That it's kind of in the world and it's kind of not in the world. It's kind of in nature, but it's not really in nature. These things are fascinating. I guess that's why also being this much closer to nature is also fascinating. I mean, this is more like the kind of Latourian labs which he describes in some of those early texts, pasteurization in France and things, like the idea of the lab going out to the setting, and that the only way one can ever truly tackle this new set of issues is to sort of, encapsulate everything that's needed in this place of instrumentation and evaluation. Yeah, I mean, I think the idea of this is to, is to make the context or the subjects reconfigure the laboratory itself. Like, conceptual art, you're not so much talking about, what the object is at the end that's created, or you're not evaluating things in that term. We tend to be evaluating things in the terms of what's interesting. Yeah. And the conceptual art is sort of a way of framing new things of interest, but not just fictions, things that are kind of quasi-real, or what I call operational fiction, things that kind of exist, but things that also have a connection to the not real. And the conceptual artists are really interested in this space, kind of creating these quasi-real things that are like instances of an idea that is novel. So then this process, yes, this could certainly be seen as obviously conceptual art practice at all different levels of magnification, whether we look at my work or the broader BioArtCamp. Under that sense, it works at any scale you want, I suppose. But in a way, also, this project of mine, I must say, I see it as just being kind of a very earnest experiment, and that I'm not even sure how I want the work framed. To phrase it, yeah. It's also just a simple kind of test, you could say. You could just say it's all this kind of asking a question and seeing what the answer is, and maybe that's enough to make a new work from it. Maybe it's not. All about the idea, and and somehow, that the conceptual art always just exists on the back of an index card. Yeah. I don't think. I think. No, but what I'm saying is more like, for example, Jennifer, she's gonna see that for her the difference between bioart and any other form of contemporary art is the ethical
consideration that the viewer and the artist more ascribe to the work.
Right. Sure.
So do you think that this challenges kind of more the traditional understanding, not necessarily as an abstract in material form, but do you think it really brings conceptual art to another threshold, or it's just a kind of extension of...
Listen, I don't think it's a smooth, I don't think it's just sort of a smooth space where no kind of threshold has been crossed, but I do think that like conceptual art uses forms because of the form's intrinsic meanings and because of the way the form resonates against the content, right? The form is chosen for specific reasons. I think this is the same way, where the form is absolutely like so loaded, right? It's like over signifying, right? It's over signifying to the point that like when we make an object out of bits of leftover cellular mass in atoms work, right? Who really cares what the shape of the thing is?
Yeah.
What really interesting is materiality, right? That you know, it's totally overcoding and overpowering any other kind of meanings you could possibly have. So I think this is a version of contemporary art, conceptual art that we have here. But it certainly does bring a whole bunch of other issues along with it. But I would say that conceptual artists have been seeking that all along. They've been speaking to work with in spaces that are so funky and loaded that we have a tough time even seeing anything that would have resembled what would formerly been called an artwork.
Paul's sample worked?
Did Zoot film?
I think, yeah, Zoot filmed it, yeah?
Yeah, we got it. It's not a great, we don't have a great picture of it because my camera isn't great and it's not, but we got
I have a great camera,
evidence.
but I guess it's already...